This cabinet is like my Narnia. It contains all of the amino acids, and so it's like the gateway into proteins. So amino acids are protein letters. There's 20 common genetically encoded ones, and over the next 20 days I'm going to take you one by one through them all. But really they have these unique properties, and then when they link up to form peptides and then proteins, those properties are going to contribute to the overall structure and function of the protein. And so you can think of proteins as kind of like charm bracelets, so all of these amino acids have this generic backbone part, and this allows them to link up, kind of like chains that charm bracelet. But then they have unique charms that stick off, and so you end up with getting these chains, like these charm bracelets. And you can imagine that if you have different charms on your charm bracelet, it'll look different and it'll act different. And so different proteins have different sequences and links of their amino acid chains, and this is going to give them different properties. And and how they fold is largely going to be influenced by the various properties of the amino acids, and also how they interact with things, how they catalyze or like speed up reactions. It's all going to come down to the primary structure, which is like the sequence of amino acids. This will contribute to secondary structure, tertiary structure, quaternary structure. Don't worry, we'll get into all of these terms. But really, I just today I wanted to just give you an introduction to how all of these amino acids are going to fit together to form proteins, and why it's important to know all the different amino acids before we go more depth into all of them. So happy 20 days of amino acids? Well, almost. So what I'm going to be talking about are the proteogenic ones, so the ones that you can actually like find in proteins. So there are also some amino acids that serve important like messenger roles and stuff, but they're not like found in proteins. You can also have amino acids be modified after they're put into proteins, so those want to be like genetically encoded. And then even these ones that we're going to talk about, so like these main 20, they can also like moonlight with other roles as serving as messengers and that sort of thing, intermediates and base in various metabolic pathways, all this cool stuff that we'll get into as we go one by one through them. So quick terminology. So amino acids, those are like the individual letters, and they get that name because they have an amino group and a carboxylic acid group, which is typically deprotonated in the carboxylate form, as we'll get into. But when they link together through these special bonds called peptide bonds, which we'll talk about. If you've taken like OChem, you might be used to having these referred to as amide bonds. So when they're linking amino acids, we call them peptide bonds, but it's an amide bond. So it's this carbonyl, so a C double bonded to an oxygen next to a nitrogen, and it has this cool like resonance. So there's like partial double bonded character, and you have electrons shared throughout. We'll talk about it, don't worry. But it has important structural consequences because that's the state of the plane. Don't worry, we're going to get all into this. I'm just giving you an overview. So when these amino acids link together to form these, you link multiple, you keep linking them up, you get a peptide. And so we typically talk about peptides when they're like short, like 40 or so less. When you get a longer peptide, so you have like a polypeptide, so lots and lots of amino acids linked together, then these polypeptides fold up and you get these like functional proteins. And so a protein is just like a big old chain of amino acids that folds up and then into unique shapes, and those unique shapes will give it like allow it to serve unique functions. And all of this is coming down to basically what amino acids are in that chain, in what order, and how long, and all of these various things, cellular conditions, all sorts of things. But it's really important to know these amino acids. Sometimes the terminology can get kind of confusing. So when amino acids link up to form peptides, so on one, we talk about them as going from the N, reading peptides or whatever, from the N terminus to the C terminus. And so the N terminus is where you have your amino group. So often it's drawn in by convention, it's typically drawn with like NH2, which is the uncharged form, but typically in the body, it is in the NH3 plus, so like the charged cationic form. And then at the other end, you have your carboxyl group. And so this is often drawn in this, in its non-ionic form, it's with like an OH, but this is going to be deprotonated in your body because this carboxyl ion is like so stable, because when you lose that hydrogen, now this can be involved in resonance. And remember, resonance is really, really awesome. And so they like having this resonance, and they're happy even though they have that negative charge, they can then share it. And so we typically draw them in their non-ionic form, but typically at like the pHs that are normal or whatever, this is going to be protonated and this is going to be deprotonated. So your carboxylic acid is your carboxylate group, and then this is a cation. If you were to go higher in the pH, so you have fewer protons available, this would deprotonate and be neutral. But this would still be negative. And if you were to go lower, so where they're in pH, so there's more hydrogens available, this would become protonated, but this would still be protonated. So either way, you're never really going to have this form where you have it totally neutral with the NH2 group and a C double bonded to an OH, so the carboxylic acid group. But that's typically how it's drawn, so don't let that confuse you. Typically, they're in like this like zwitterion form. So if you had like a free amino acid, it would be in the NH3 plus and the COO minus. And so those charges cancel each other out, so it's neutral overall. And so we call it a zwitterion, which is a really cool word, where you have like a positive charge and a negative charge, but neutral overall. When you link up amino acids to form a peptide, then you're only going to have one free amino group. So this would be the N-terminus, N for the nitrogen, and then one free carboxyl group, so C terminus, C for the carboxyl. And then you've actually, in the process of joining these together, you've lost the equivalent of a water, so we can call this like a condensation reaction, although that's not how it happens in our bodies, and I have more on that in a post if you want to read that. But then you can also break it by adding water, so hydrolysis. So hydrolysis can break these bonds, but you typically need like acid and stuff to do that, because these bonds are really, really happy. These bonds are really strong. All has like partial double bonded character, and double bonds are really strong. And so this is like a strong bond. When they also join up together, so now you have basically, you have the you have this backbone, and then you have the side chains sticking out. The side chains that stick out are the parts that have the unique properties. We typically, so because they've lost the amino group and the carboxyl group, basically when they join together, so this was a part of a carboxyl group, and this was part of like that NH3 group. But when they join together, now you don't have that. And so now we call the leftovers like residues instead of amino acids, although we often just like interchange the two. But technically these are now residues because they're not amino acids, because they've lost like their acid part and stuff. We also call these refer to these as alpha amino acids, because like the bonds and stuff are in the side chain, and it's all coming off of this central alpha carbon. So a protein has a ton of carbons, but the alpha carbon is like that central hub where it's going to be attached to the side chain, to the nitrogen, to another carbon, and to a hydrogen. And that is our alpha carbon, and then like you can imagine like if you had your amino attached to like the one next to it or whatever, it would be a beta. But in all these amino acids that we're going to talk about are alpha amino acids, and typically in the L stereoisomer. You link up amino acids to form peptides and get enough of them, and they fold up and you get a protein. Well, what you really have is in this polypeptide is like a chain of planes, and so these planes, so what are these post-it notes hiding? They're hiding this peptide bond. So this peptide bond involves this nitrogen, this carbon. This carbon is double bonded to this oxygen. And these are stabilized by resonance. So basically they're sharing electrons between them. So when atoms, like all these are representing individual atoms, and so the color is representing like their identity. So this is a nitrogen, this is a carbon, this is an oxygen, and the white ones are little hydrogens. So these are all joined together by sharing pairs of electrons in what we call covalent bonds. Share one pair, you get a single bond. Two pairs, you get a double bond. And so this carbon, you can't really see it when you have a model like this, but this oxygen is double bonded to this carbon. But and same with all these ones, but the rest are single bonds. But basically what can happen is that in this bond, although we draw it as like the carbon being double bonded to the oxygen, this whole bond is actually resonance stabilized, which means that those extra electrons are actually being shared among all three of these atoms, the nitrogen, the carbon, and the oxygen. Now, this can only happen if the three of these lie in a plane. So these have to be in a plane. You can't have them... These have to be in a plane. You can't have rotation around this bond. That's a no-no. And so that's why you end up like with this chain of planes, where there are only certain places where you can have rotation. So in a model like this, you can rotate these bonds, but in reality, you can't really. But you can rotate all the other bonds. You can rotate the bonds around these, which is why you get like this chain of planes, where these peptide bonds, these can't be rotated, but you can rotate around them. And we have specific names for these different angles that you can get when you rotate around these bonds. When we talk about rotation around the nitrogen and the carbon, this alpha carbon where the side chain's hanging off, this rotation is called the phi angle. So it's like Greek letters, like a stick with a circle. We can also have rotation on the other side of this plane, and in this case we have between the carbonyl carbon and the alpha carbon, so on the other side. And so this rotation, this is called the psi angle. And so this one is like a stick with a C. And so you can think of C for carbon. So remember, the psi angle is between the carbon and the carbon. And by the angle we're talking about is going to be like the angle between the plane formed by them. And so you don't have any rotation between between these three. So between the carbonyl carbon and this oxygen and nitrogen, you don't have rotation here because this has this partial double bond character due to the resonance. But you have rotation on either side, and this rotation is going to be more free than this rotation, but it's going to be restricted by like having steric clashes and stuff when things are coming into contact with other atoms and being not happy and stuff, and some things like hyperconjugation we won't get into. So you end up with this chain of planes, and you end up with this restricted motion of the backbone, and this is going to help give proteins their shape. What's also going to help give them their shape is that these oxygens and the nitrogen, so these are the generic parts that every amino acid has, these generic parts. And these generic parts are going to be able to hydrogen bond. So oxygen and nitrogen are both highly electronegative. This means that they're going to pull on the electrons that they share with other atoms, unless those other atoms are more electronegative. But in these cases, the oxygen is more electronegative than carbon, and the nitrogen is more electronegative than hydrogen. This means that when an oxygen is bound to a carbon, the oxygen is going to be pulling more electrons towards it. Those electrons are negatively charged. This is going to make the oxygen partly negatively charged. What about the hydrogen? When you have a hydrogen bound to a nitrogen, the nitrogen is going to be hugging the electrons. This makes the nitrogen partly negative, the hydrogen partly positive. You have something that's partly positive, you have something that's partly negative. If you get these lined up in such a way that they can come together, then you're going to get you can get hydrogen bonding. And so this isn't a very realistic typically they're from farther apart unless you're like in a turn or something, but I am having a hard time orienting my model because it's too willy-nilly loosey-goosey. But the basic idea is because you can have these hydrogen bond donors from the hydrogens on the nitrogens, and the hydrogen bond acceptors on the from the lone pairs of electrons on the oxygen. And so when these can come together, you can have different parts of the protein interacting with one another through those backbones, through hydrogen bonding of the backbones. And this is how we get secondary structure or motifs. So these like basic folds that come up a lot, such as alpha helices, where you have these turns where there's like hydrogen bonds between like the first and the fourth, and then a turn and the right-handed turn. You can have beta strands, which are like long strands that are kind of like zigzaggy, and then they form these beta pleated sheets, like these pleated sheets things. And so all of that is involving these hydrogen bonding of the backbone. And that's just involving the generic part of the backbone, which all of these amino acids have. But then each of them also has the unique part, and it's these unique parts that are going to give the protein, make the proteins kind of like different. So in addition to helping dictate what type of folds the protein will take, so you can imagine that some of the types of folds, like if you're trying to make it like a helix and you have some like big weird thing, that's not going to want to be in a helix. So the side chain can help influence what structure it takes by restricting the angles that the backbone could take. And remember, when we talk about the backbone angles, we're talking about the phi and psi angles because we're not rotating around inside that peptide bond. We're not going to rotate that, so we rotate around them. And so there are certain places where we could expect to find like certain like preferred angles of the phi and psi, and we can plot these on like a Ramachandran plot. And the tendency for different amino acids to be found in different ones of these structures can be determined in part by like how bulky and that sort of thing they are. And we'll get into all of this more as we talk about the individual amino acids. But when they adopt these folds, so they get their secondary structure based on just the backbone. But remember that the angles that the backbone can take is restricted both by the peptide bond, which is generic, and by the side chains, which are unique to the amino acids. What's also unique to the amino acids is the binding opportunities offered by those amino acids. So if you have something like a glutamate, where you have like a negatively charged group, that can react with something like a lysine, which has a positive, to form like a salt bridge, or like a charge-charge ionic interaction. You can also have it interact with like partial charge things. And a lot of times these things are really just like interacting with water because water's everywhere. But when you get interactions between the protein side chains, like between different protein side chains in the same protein, that can help provide the 3D structure of the protein through what we call its tertiary structure. So the tertiary structure is now you get the side chains involved. Secondary structure, we're talking about the backbone, but remember that the angles that the backbone can take is restricted by the peptide bond and by the specific amino acids side chains. But the tertiary, now we're getting the side chains involved, and so these can bond to other parts of the protein and that sort of thing. And they can also like bond to the backbone and that sort of thing. So, and then quaternary, that's where you evolve like other protein chains. So a lot of times we talk about proteins as just being like this single chain that folds up to form this nice protein, but often proteins are working as part of like a multimer. So you have like a dimer where there's 2 proteins, or a trimer where there's 3. So these can be 3 copies of the same chain, in which case it would be like a homotrimer, or they could be like different chains, which would be like a heterotrimer. So when you get different protein chains interacting, then we call that the quaternary structure. And we can get a look at all of the fancy 3D structures that they make using techniques like cryo-electron microscopy, or cryo-EM, and X-ray crystallography. And that's where we get all those pretty structural models that you see. But those types of interactions are all involving the same types of things. So whether it's between this part of this protein and another part of this protein, or whether it's between part of this protein and another protein chain, those are all involving the same types of interactions that we're going to talk about, which are like these, we can talk about like hydrogen bonding, we can talk about salt bridges, hydrophobic interactions, all of these various types of interactions that we'll get into. But there's different interactions offered up by different side chains because these side chains are all unique. And the side chains are also going to influence, so the side chains are going to influence how the protein interacts with other parts of the protein, and also the basic protein's overall fold based on something called like its hydrophobicity or its hydrophilicity. So we'll talk about this more in detail, but basically remember that when we're talking about like cytosolic or like water-soluble proteins, so the ones just hanging out in our cell, we're not talking about the membrane proteins here. So there's water everywhere. The water is really polar, so oxygen, remember, is really electronegative. So it's going to pull on the electrons and make the things attached to it partly positive. So when the oxygen in water is attached to two hydrogens, the oxygen is going to pull the electrons, become partly negative, the hydrogens are partly positive. So water can do that like hydrogen bonding stuff we were talking about in the peptide backbone. In fact, water is like the best hydrogen bonder ever because it has like 4 opportunities. You have the 2 donors from the hydrogens, the 2 acceptors, the lone pairs on the oxygen. So water forms these really form like these networks of hydrogen bonding. And so water really likes to hang out with itself because it's so good at hydrogen bonding with itself. But if you have some of the amino acids, so some of like the side chains offer opportunities for hydrogen bonding or even like full charge to partial charge interactions. But some of them are, and we call those hydrophilic, the ones that have things that can form like these attractions with the water. But then we have things that are like hydrophobic, so like non-polar amino acids, and these don't offer anything for the water. So when you have this sort of thing, then the parts that the water likes will hang out on the outside, and the parts that the water doesn't like, the hydrophobic parts, will end up clustered in the middle. So the main effect, the main like driver of protein folding is actually this hydrophobic effect, where the protein is situating itself so that it's minimizing, the hydrophobic parts are minimizing their contact with the water, so that the water is happier because it doesn't have to be forming like these weird clathrates or like water cages, like trying to like cordon off the hydrophobic parts of the protein. You can also imagine that if you have a membrane protein, then the hydrophobic parts are going to be on the outside facing the membrane, and then the hydrophilic parts would be like on the either end, like facing into and out of the cell, or like in the center of a pore and that sort of thing. So hydrophobicity and hydrophilicity is going to be a big thing, and we'll talk about the relative hydrophobicity and hydrophilicity of various amino acids as we go through them, as well as things like their size. Their bulkiness is going to affect how they can turn. So we talked about how like big bulky ones might not want to be in certain structures, and when we talk about bulk, that bulk can be like at the end or it can be in the center. And so when we have like branched amino acids where they have like this bulk, something like valine, it's going to have a bunch of bulk close to the backbone, and so that's going to really restrict its motion. Whereas something that has like, it's long but it has its bulk at the end, well, that's going to have less restricted motion. And so there are all sorts of different things about these amino acids that make them really unique and exciting. If you're like taking biochemistry or that sort of thing, I suggest that you memorize, at least if you can't memorize the structures, or you don't want to memorize the structures, if you don't need to memorize the structures, whatever. But I mean, it's good to know that, but what's really, really important is to know like the various functional groups in the side chain, so like the special parts about them, and the special properties about them, like know which ones are hydrophilic, know which ones are hydrophobic, know which ones are positive, which ones are negative, which ones are big and bulky, which ones are small, and be able to recognize them if you see them, even if like you couldn't off the top of your head draw out a tryptophan. But you should also be able to recognize, so when we talk about amino acids, we have like they have like their full name, and then they have like an abbreviated name, like their nickname, I call it, which is like a 3-letter code, and then their like initial, which is a single letter. So we have a single letter code, a triple letter code, and then their full name. And so it's really important that you learn the names so that you can identify them. So some of them are kind of easy. So like glycine is G, but then when you get to glutamate, well, G was already taken, and so now glutamate is an E, and then you're like, what the heck, why is glutamate E? Well, because glycine took the G, right? So you need to know these so you can recognize them, because it's a lot easier when you're dealing with huge old proteins in order to use like a 1-letter code in order to not have like super duper long things if you were to write out every amino acid name. So you want to be able to use these 1-letter codes, but then you have to make sure that people know what those 1-letter codes are. So I highly recommend that you learn them, and I'll go through them all as we go through the amino acids one by one. And we haven't even talked about the fact that when these proteins fold up, they're gonna fold up, like they can have, we've talked about proteins interacting with like themselves and with other chains, but they can also interact with other proteins, and they can interact with other like substrates and stuff. So they can actually like form the active site of enzymes. So enzymes are like reaction speeder uppers, and they're often proteins. And so basically, if this big protein folded up, and all of this protein, so all of the backbone, all of these side chains are helping contribute to this protein's full overall shape and fold. But then like the actual reaction that's happening, it's happening in this tiny little pocket in there, which is like perfectly formed and situated for this reaction to occur. And then like in that active site, you're going to have like an amino acid residue sticking out there and able to like act to help accelerate these reactions. So we'll talk about some of the various things like serine proteases, cysteine proteases, all of these really exciting things, and these ways that these various amino acid side chains are helping make life happen. And all of this is happening in the context of this larger protein, but a lot of that larger protein is really just kind of like helping determine the fold, as well as like other binding interactions and stuff. So typically well, I talk about like the whole protein being involved. Well, often proteins have like structural domains. So these are parts of the protein that you could basically chop off the rest of the protein, and this part would still fold up, and in the same way it would as if it were in the full protein. And so we call these like structural domains. And it's really helpful for like structural biology and biochemistry and stuff if we know where those domains are, and so then we can like chop them apart and investigate them more easily individually, and more likely to get like a crystal structure of them and stuff. But we can also talk about like functional domains. So basically that doesn't necessarily mean that you could cut those parts of the protein apart, but it does mean that like that has a function, like a defined function. So maybe it's like, you can talk about if you have like a studio apartment and you have like one little side of the room that's your kitchen and one little side of the room that's your bedroom. You can't really cut those apart. You can't really cut your room apart and have them still be a room. Everything would just like fall out, right? But you still have distinct functional areas. Whereas if you had like two separate rooms, like a kitchen and a bedroom, then those would be like structural domains because you could chop them apart and you still have separate rooms. Yeah, that's my weird analogy.
The order that these amino acids are added is determined by the messenger RNA, which is determined by the gene. So basically the gene is the DNA version of the protein recipe. And then that gets copied into this RNA form, this messenger RNA. There's also a little editing that goes on to remove some like regulatory parts called introns. But then you get this mature messenger RNA or mRNA. And then this complex called the ribosome, which is like this protein RNA complex, is going to help connect the amino acids together in the order that's defined by the mRNA. So basically it goes along and it reads this mRNA in 3-letter chunks, where each of those 3-letter like words are called a codon. And so each 3-letter combination will spell like one amino acid. And so the ribosome goes along, and then there's like this connector molecule between that brings the correct amino acid. And so this is called a transfer RNA or a tRNA. So basically it's an RNA like it sounds. So it can form like RNA base pairs, just like normal RNA could, except in this case, in one part of, so this RNA is all big and folded up inside this kind of like L thing. And then on one end, it has this like anticodon that's going to match the codon. And then on the other end, it's going to have the corresponding amino acid. So it's going to have an M if it says, if this codon says to this spells N, then it'll bring an M. And so this will match and this will match. And then it comes along, and in this process of translation, it's going to bring the complementary one. Then the ribosome is going to help link them up. And then some like elongation factor things are going to come and help kind of like shift things along. Then the next tRNA can come. So the ribosome shifting, now it's at the next codon. So remember, it's moving 3 letters at a time, because each of those 3-letter mRNA words, these codons, is going to spell a single amino acid. So it does this, it does this, it does this, and then eventually it gets to a stop codon. So this is a codon that spells stop instead of spelling an amino acid. And then like a release factor comes and like cuts things off. And now you have this big peptide. And this big polypeptide can fold up to form a protein. So typically though, it's like it's folding like as it's coming out of this ribosomal tunnel. So what happens if you have a part of the protein coming out that is like hydrophobic or needs to bond to a part at the other end of the protein that hasn't come out yet? So there are actually proteins called chaperones that can kind of like help kind of hide the parts of the protein as they come out, and prevent them from just like clumping up and aggregating until the rest of the protein is like ready to fold. And so this can like help keep those hydrophobic parts happy, keep everything happy until the protein can like fully fold. So sometimes, and this is one of the reasons why proteins might have a hard time, you might have a hard time recombinantly expressing proteins. So basically, when you put the instructions for making the protein into like like a vector, so a vehicle such as like a plasmid for getting them into cells that can express it for you, like bacteria. So sometimes bacteria don't want to make things for you because they don't have all the same chaperones and stuff, which is, I mean, there's multiple reasons, such as like post-translational modifications and all sorts of stuff. But one of the reasons is that like sometimes your proteins need these like chaperones and stuff that are found in our cells, but not in the bacterial cells, that really help make things fold properly. Yeah.